And are we good to go now? Okay, great. But we don't have the slides up anymore.
Just unshared. Let me share again.
Yep. Okay. I'm just going to introduce you, Simon, and then we'll get going. So we're in a seminar room, so we have like a hybrid thing. People can log in online, but also we're in a seminar room as well. So there's a bunch of people here listening as well.
I'll be careful what I say then.
Sorry?
I'll be careful what I say then.
Yeah. All right, so I will get started. I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to the elders past and present, and the elders from other communities who may be here today. So today it's my pleasure to introduce Simon Stott, who's the Director of Research at Cure Parkinson's. Cure Parkinson's is an international funder of research focused on disease modification for Parkinson's disease. The charity supports both preclinical and clinical studies attempting to slow, stop, or reverse Parkinson's. Simon is originally from New Zealand, and he has over 20 years of experience in the field of Parkinson's research in both academic and biotech sectors. He's been involved in lab-based research as well as clinical studies, and is co-author on over 50 publications. Scientific publications. He also maintains the Science of Parkinson's website and also a very active Science of Parkinson's Twitter feed, which I myself have found very handy to keep up to date with all the latest Parkinson's research development. And I actually must comment, Simon, that it's very insightful tweets. Like, you really get to the bottom of the science and explain it very clearly. So that's very handy for many people in the space. So thanks, Simon. Whenever you're ready to begin, go ahead.
Thank you, Michael. That's the nicest introduction I've ever had, I think. That was beautiful.
Thank you.
So hi, everybody. My name's Simon. I'm the Director of Research at Cure Parkinson's, as Michael was saying. And my only issue at the moment is that the slides are not moving forward. All right, hang on, let me stop sharing, and we're recording this, aren't we? This is embarrassing. Let me try that again.
That's all right.
And this time we're moving, yes. So my only disclaimers are that I'm an employee of Cure Parkinson's, and as Michael was saying, since 2005, the charity has been focused on disease modification for Parkinson's. I'm going to be talking about a few studies that we've been directly funding. Where that is the case, I have just put our label, our logo for reference. So what I'd like to do is start off by trying to put everything I'm about to say into context. And it basically begins with this chart here, which looks at all the scientific research that's ever been done on Parkinson's since James first described the condition. And what you notice is there is a bit of a hockey puck of a graph here. And over 80% of the research has been done on Parkinson's since the year 2000. But when historians go back and write the history of this condition that we know of as Parkinson's at the moment, they will hopefully see 1997 as a turning point in the war. This was the year that Dolly was introduced to us and Diana was taken from us. And Leonardo DiCaprio and Kate Winslet were on the big screen on Titanic. But it was also a year that a small region of DNA became really important to the Parkinson's field. In 1997, a group of researchers from the NIH in the US, they were looking at a Italian family, and they noticed that there was a very high incidence of Parkinson's. And they took DNA from the greater extended family, and they looked for where there were variations in those individuals that were affected by Parkinson's. And they found a group of variations in the alpha-synuclein gene. And what's really amazing about this story is that just 3 months later, Maria Spallanzini and colleagues at Cambridge University and also at Pennsylvania University found that alpha-synuclein labels Lewy bodies inside the brains of people with Parkinson's. Lewy bodies being these dense, circular, spherical clusters of aggregated protein. So all of a sudden, alpha-synuclein became the boogeyman, the public enemy #1 of Parkinson's. And over the next few years, there was this gold rush of geneticists looking for other genetic variations that could be associated with Parkinson's. And 1 year later, they found Parkin. A couple of years after that, they found variations in DJ1 and PINK1 and LARC2. And now we have over 80 genetic regions associated with Parkinson's. So the early 2000s were a really busy time for establishing the genetic variations associated with the condition. And then the researchers started looking at the biology associated with those genetic variations. And they started seeing signs that pointed towards the pathophysiology of Parkinson's. And obviously there were opportunities there to start thinking therapeutically. For example, genetic variations in the GBA1 gene. This is one of the most common genetic variations associated with Parkinson's. They found that these genetic variations, quite a few of them, were associated with reduced activity of the enzyme that's associated with that gene. And so the 2010s was this really busy period of establishing the associated biology and then developing tools to look at the pathways involved and also doing drug screens. And so now in the 2020s, we're at this really exciting period where biotech companies and pharmaceutical companies are lining up clinical trials that are based on all of this associated biology. And they've identified really solid targets that have been drugged now. And we're moving towards clinical trials that could potentially be slowing or stopping the progression of Parkinson's. So that just hopefully puts a bit of context to what I'm about to talk about. And what I'm going to do today is just go through a couple of the more exciting areas of research currently focused on disease modification for Parkinson's. And to do that, I'm going to start off with alpha-synuclein. So alpha-synuclein is one of the most common proteins in the brain. It makes up about 1% of everything that's up there. And we're still investigating exactly what it does. It has a lot of different functions, primarily around the synapse. But what happens in Parkinson's is that it starts to aggregate in neurons in vulnerable areas of the brain in people with Parkinson's. And one theory as to how Parkinson's could be progressive is that this aggregated alpha-synuclein could be being passed from cell to cell. And so researchers have decided that one way of slowing down the condition potentially is to grab that alpha-synuclein as it's being passed. And to do that, they've designed immunotherapies based on antibody approaches to grab that alpha-synuclein as it's being passed from cell to cell. And there have been two main clinical trial programs focused on this. One of them is, let me just get rid of that. One of them was conducted by the biotech company Biogen, and the other one was conducted by Roche. They both had active immunotherapy approaches to targeting alpha-synuclein. And after 12 months of testing different doses in very large cohorts, they both found absolutely no difference between placebo or any of the doses used. So Biogen decided, right, we're out of here, we'll leave. But Roche went back and they had a look at the data because they thought they were seeing some interesting trends. And just recently, the team at Roche published this paper just a couple of weeks ago, actually. So what they found was that their immunotherapy called prasinezumab has positive effects in terms of very fast progressors. And this is just after the 1 year data. And so then they went back and they had a deeper dive into the data. And while there was no difference when they looked at their primary endpoint, which was UPDRS part 1, 2, and 3, this is a clinical rating scale that's used as the gold standard for tracking the progress of Parkinson's. Well, they found no difference between the two doses, which are in black lines here, and the average of them is in red, versus placebo, which is in blue. When they look at UPDRS Part 3, which is the motor part of the rating scale, and it's assessed by clinicians as opposed to the patients, they see that there is a reduction in the progression of motor symptoms between both groups of treated individuals versus a placebo. And they also speculate that there might be a plateauing of progress towards the end of that treatment period as well. So there's a bit of a slow start, but then maybe the participants really slow down in the progression towards the end of that year. And so they set up an open label extension study to continue looking at this. And this is just called the Pasadena Open Label Extension Study. And it involved 52 weeks of everybody being on drug, but different doses. And then a further up to 60 months of everybody being on the low dose of prasinezumab. And one detail just to point out here is that in the middle of between part 2 and part 3, there was this small event called COVID, which sort of disrupted things a little bit. So just keep that in mind when you consider the data that we're about to look at. But what they found in that long-term extension study, so everybody is now 4 years plus, and they found that everybody, once they shifted to treatment, basically plateaued compared to the Michael J. Fox PPMI study control data that they were using as their control in the open label extension. Not only on their part 3 motor scores, but also on the part 2 scores as well, which are judged by the participants themselves. And one other detail just to note here, across the bottom of the screen, you can see the number of patients. Prasinezumab requires monthly treatments. So for 4 years, these individuals were prepared to get monthly treatments and stay part of the study. So that the participants obviously thought that there was something worth continuing with here. There wasn't a large dropout. When you look at the early stage numbers, you move from 177 participants to 166 at 4 years. That's a pretty low dropout number. So this is quite encouraging. But the team at Roche are very quick to point out that all of this is post hoc and open label extension data. So it's not properly controlled, and they're very cautious about making too much noise around this. But what they have done is they've set up another clinical trial. It's 18 months, and it's involving 586 participants called the Padova study. And that's due to complete in September of this year. So it'll be very interesting to see that data come hopefully early next year. And if you're interested in more on this, there's a video of the Roche team presenting the data down the bottom there. I'll take a moment to let you snap a photo of it. But if you want a copy of these slides, just reach out and tell me. There's nothing confidential here, so I'm happy to share slides. Now, the immunotherapy approaches are not the only path being taken for alpha-synuclein. Immunotherapy antibodies don't do a great job of getting into the brain, let alone cells. So biotech companies are focused on small molecule inhibitors of alpha-synuclein aggregation as well. And the most advanced of these are these two studies. Firstly, UCB and Novartis have paired up to conduct an 18-month study called Orchestra, which is looking at UCB 05599. In 450 participants with early Parkinson's. The primary completion date of that study is April. So that's completed now. And hopefully we'll be finding out the results of that study later this year. And then there's also a study by a company called Anovis, which has conducted a short and very small phase 3 study with an old molecule called Posifen over a 16-month period. It finished late 2023, and hopefully we'll know the results of that study soon. So now what I'd like to do is shift attention to a different approach that people are taking. focused on a molecule called LRRK2. So this is a protein or leucine-rich repeat kinase LRRK2. It's a multi-domain protein. It has a lot of different roles in cellular biology, similar to alpha-synuclein. But key among them is the kinase domain that you can see along the bottom there. Approximately 1% of people with Parkinson's have genetic variation in LRRK2. What I should add is that just because I'm talking about all these genetic variations associated with Parkinson's, it doesn't mean that Parkinson's is a genetic condition. Only about 15 to 20% of the overall Parkinson's population carry genetic variations. And it's important to point out that there are lots of non-presenting carriers as well. So we can't say that if you've got one of these genetic variations, you're definitely going to go on to get it. The penetrance is variable at best. But LRRK2 associated Parkinson's is a slow progressing form of Parkinson's. And what it results in is a hyperactive version of this kinase. And so biotech companies such as Denali Therapeutics have been exploring inhibitors of LRRK2 activity. And Denali's really been leading the charge here. And in 2021, they announced the results of their phase 1 testing of their molecule DNL151. They tested different doses of the drug in healthy individuals, as well as 36 people with Parkinson's over 28 days. They found it was safe and well tolerated. And they also found that it basically did what it was said on the can. And the left-hand graph here, you can see across the middle, a gray line, that's the control participants. And all of the colored lines are different doses of the drug on LRRK2 activity. So you can see once they start treatment, that LRRK2 activity really does crash down towards 80% or 75% reduced activity. But it's not only LRRK2 that's affected by the drug, also the downstream targets. In this case, RAB10, they see significant reductions in RAB10 activity as a result of treatment with these inhibitors. So that was very positive. In fact, positive enough for Biogen, the biotech company, to step in and say, hey, we'll partner with you in the co-development of this. And in 2022, they initiated the LUMA study. This is a phase 2B clinical trial of the inhibitor in 640 participants with both idiopathic Parkinson's and LRRK2 associated Parkinson's participants. It's a 2-year study, and it's expected to complete in late 2025, early 2026. So that'll be something to look out for over the next 18 months. Now, one question you could ask is increased LRRK2 activity occurring in people with Parkinson's, with idiopathic Parkinson's, excuse me? And could these LRRK2 inhibitors be useful in treating this broader cohort? And the data suggests yes. A group in University of Pennsylvania, led by Timothy Grenemeyer, did a large post-mortem study of people with idiopathic Parkinson's, and they found elevated levels of LRRK2 in vulnerable regions of the brain. And Denali also state that in the idiopathic cohort of their phase 1 testing, they see RAB10 levels almost 2 times the higher in the sporadic Parkinson's individuals as opposed to the unaffected healthy controls. So there is certainly evidence to suggest that this treatment approach could be useful in the broader Parkinson's community. And it's a very busy area of activity as a result. There's a lot of biotech companies exploring this space. Merck and GlaxoSmithKline, 2 large pharmaceutical companies, have very large programs focused on this. And there are 4 companies, in addition to Denali and Biogen, that have clinical programs, and I've put them in red squares here. Of interest is Avinas, who are currently exploring phase 1 testing, currently conducting phase 1 testing on a PROTAC for LRRK2. So that'll be one to keep an eye on as well. I'm going to move again to a different one of these, a different pathway. and that's glucocerebrosidase. This is encoded by a gene called GBA1, and as I said before, GBA1 is one of the most common genetic variants associated with Parkinson's. And as a result of variations in GBA1 gene, you see a reduction in levels of GCase protein. This is glucocerebrosidase, which is a lysosomal enzyme, and it's involved in waste disposal of cells. And people with Parkinson's, even idiopathic Parkinson's, have reduced levels of GCase in their brains. So researchers have been exploring activators of GCase activity. How can we raise levels of this activity, and can that actually have an impact on the progression of Parkinson's? And one of the lead molecules here has been ambroxol. This is a respiratory medication, a cough medicine that's widely used in Europe. It's not registered in the UK or in the US or in Australia, as I understand it. But drug screening studies found that it elevated levels of GCase in cells, and it also had neuroprotective effects in models of Parkinson's, both traditional neurotoxin models, but also transgenic models, and it elevated glucocerebrosidase levels in non-human primates. So a few years back, Cure Parkinson's and co-funders Van Andel Institute and John Black Foundation funded a phase 2A study looking at ambroxol in people with Parkinson's. We took 18 participants, half of whom actually had GBA variants, and we treated them for 6 months. And this was a really heroic effort, because for 6 months, these individuals took 22 pills of ambroxol per day, in addition to their dopamine medication. And the pills have a really bitter aftertaste, not on the tongue, but sort of on the back of the throat. So it's not exactly nice stuff to be taking every day. But after that 6-month period, everybody on the study had higher levels of GCase activity in the CSF, GCase protein in the CSF. One of the challenges for the field is actually measuring GCase activity. But there were higher levels of GCase protein in the CSF, and there were also signs that there could be some clinical benefit. It's an open label study, so you have to be careful with interpretation of that data, but it was encouraging enough to lead forward to a phase 3 study that's now being set up. And this is being led by Professor Anthony Shapiro at UCL here in London. And it's going to be a 2-year study involving 330 participants, half of whom will have GBA1 genetic variants. And the primary outcome will be UPDRS parts 1 through 3. And again, you could ask the question, could a GCase enhancer be useful for idiopathic Parkinson's? And the data suggests that yes, it could be. Research has shown that there's generally a reduction in GCase activity in the brain with normal aging. And here you can see in the substantia nigra and the putamen, two areas that are badly affected by Parkinson's. You can see a general trend towards lower GCase protein levels. But in people with Parkinson's, this seems to be exaggerated. Sporadic Parkinson's here is labeled in the dark bars. And through the 60s and 70s, you can see that these individuals have much lower levels of GCase activity. So there is a strong or a compelling argument for the use of GCase enhancers in idiopathic Parkinson's as well. And that's why there's this idiopathic cohort in the ASPRO phase 3 study. But ambroxol is not the only molecule that's being explored here. And this is a really busy space in terms of clinical research. Similar to LRRK2 inhibition, there's a lot of biotech companies exploring this. Some of them are going with small molecules, others are going with gene therapy approaches. And again, all of the companies on the screen here that are boxed in red have got active clinical trial programs. One of the Lilly and Prevail is actually a gene therapy approach, and that's been going for a couple of years now. Again, I'm going to move this time away from the genetic risk associated therapeutic investigations towards something more down to earth. And that's GLP-1 receptor agonists. So GLP-1 receptor agonists are or glucagon-like peptide 1 receptor agonists are used in the treatment of phase 2 diabetes. And it's been a molecule of interest for us at Cure Parkinson's since 2011, when we were supporting a lot of preclinical research on it. And in 2013, we funded a open label phase 2A study at UCL in London involving 44 people with Parkinson's. And they were compared to, half of them were on drug. We couldn't get the placebo at the time. So it was basically an open label study exploring the GLP-1 agonist exenatide versus the standard treatment. And exenatide on the graph on the left here is the dotted line, and it's UPDRS part 3, so the motor score is in the off state, so they're not on medication at this point when they're assessed. And as you can see, the controls continue to progress, while the exenatide treated group improved a bit and then just basically stabilized across the 12-month period. And this was present even after washout at 14 months. And then we funded an extension study to look at to follow these individuals off medication for the next 12 months, off exenatide for the next 12 months. And we found that the benefits were sustained there as well. So this was really encouraging data that stimulated a phase 2B study, which involved 60 individuals, 30 on medication and 30 off medication. 30 on exenatide and 30 on placebo. So it's a pretty uncivilized hour here in the UK. And last week, my local football team, Ipswich Town, was promoted to the Premier League. So there's still a hint of hangover. So I'm not on my best performance. In 2017, the results of the study was published. The study was funded by the Michael J. Fox Foundation. And as you can see, over the 48-week treatment period, the placebo-treated group in the red line, they continue to progress in their Parkinson's, whereas the exenatide group have a 2-point reduction on average, and some individuals had a lot more than that. But we had some responders and some non-responders. And so we see a stabilization, an improvement and a stabilization of the motor symptoms in these individuals in the off state. And at 48 weeks, there's a washout period through to 60 weeks, and you can see them starting to gradually head back towards baseline at that point. So this is a very encouraging double-blind, placebo-controlled result. And we set up 2 other studies, one looking at GLP-1 called liraglutide. And that study, liraglutide doesn't access the brain very well, but what we saw was improvements in non-motor symptoms there, suggesting that this class of drugs is doing something interesting in the bodies of people with Parkinson's, if not the brains. And just recently, we've had the phase 2 clinical trial results from a lixazenatide study, another GLP-1 agonist. This one is brain penetrant. And this was a much larger study than the exenatide study. It involved 156 people, and they were treated for 12 months. They were at 1-to-1 ratio of drug versus placebo. And importantly here, it was conducted in France across 22 research centers. The first 2 GLP-1 studies were all of the GLP-1 studies up to this point were single center studies. So what you can do here is you can take out any potential investigator bias with 22 centers involved. And what they saw here was that while the placebo group progressed, as you can see on the bar graph on the bottom of the screen here, the placebo group progressed in their part 3 motor score by 3 points. The lixazenatide group hardly moved. If anything, they improved in their motor score. And I was talking about responders and non-responders before. What they saw in the data here was that the individuals under 60 were driving the bulk of the result. where they see a much more significant result in the individuals in the lexazenatide treated group are under the age of 60 as opposed to over the age of 60. So this is all very encouraging stuff. It's not an entirely positive story. There was a biotech company that was developing their own GLP-1 agonist. This is a pegylated version of exenatide called NLY-01, and they missed their primary and secondary endpoint. So this was a study involving 250 people treated for just 36 weeks. But what they did see was when they did post-hoc analysis on the data, again, they see this positive trend in the under 60s subgroup as opposed to the over 60s subgroup. So we've got an interesting bag of data being generated by GLP-1 agonists and in people with Parkinson's, and we're slowly teasing apart who might respond and who might not. And hopefully in the very near future, we'll have more data, because at present we have the phase 3 clinical trial of exenatide, which Cure Parkinson's has been supporting here in the UK, coming to a conclusion, and we should know the results of this in late 2024. It's a 2-year study involving 200 participants with Parkinson's. It's taken place over 6 different sites across the UK. And at the same time, there's been a phase 2 study in Stockholm, which has involved 60 participants treated for 12 months. Actually, that should be 18 months. That's an 18-month study, this one. And what's interesting here is that they've done a lot of brain imaging analysis as well. So, and we're hoping that those results will be available come mid to late 2024 as well. So there's a lot of activity in clinical trials looking at GLP-1 agonists in the context of Parkinson's. And before I finish, I just wanted to add one more thing. There is a large program here called EJS, Edmund J. Safra ACT-PD, or Accelerating Clinical Trials for Parkinson's. One of the big issues with conducting clinical trials for Parkinson's is it's a bit like building a stadium, a football stadium, playing a single match, and then deconstructing the football stadium. And so you get this silly situation with clinical trials that we set them up, we conduct the trial, we dismantle them, and then we start setting up the next clinical trial for the next therapeutic agent. And as a result, it's a very slow and expensive and cumbersome process, and has very little, it's not the most efficient approach, particularly for the patient community. And so what would be a better approach is just to have a constant conveyor belt that is continuously testing new experimental therapies. This would be a much better design. And that's where the EJS ACT-PD approach comes in. This is a multi-arm, multi-stage clinical trial platform. In Australia, you actually have the Australian Parkinson's Mission, which is a multi-arm study. But in this case, it's going to be multi-stage as well. So with the ACT-PD program, you'll start off with a single placebo arm and multiple treatment arms. And there'll be continuous assessment and analysis of the data. Instead of waiting to the end of the trial, you'll be analyzing the data in the middle of the trial and looking for which treatment's working and which one's not. And so after a couple of years, you might realize that treatment 2 and treatment 3 are not working. So let's take them out and put 2 new treatments in. Treatment 1 actually looks pretty good, so we'll continue with that. And this can continue, and it becomes quite a dynamic and adaptive process, but it's more importantly, it's just continuous. So treatment 1 here will go from phase 2 into phase 3 and all the way through to regulatory approval if it's successful, as opposed to doing a phase 2 study and then pausing, analyzing the data, waiting 12 to 18 months to set up the next study and get funding for it. It'll be a much quicker, more accelerated form of testing disease-modifying therapies for Parkinson's. And this platform will be focused on disease-modifying therapies for PD. And there's also the opportunity for industry to come in here as well and test their agents once the platform is up and running. And so at the moment, there's a large consortium of PD researchers, people with Parkinson's and care partners and other stakeholders, all voluntarily collaborating in large working groups to plan out how this project will get started. And we're at the point now of trying to secure the funding for it. So this is something to keep an eye on in the not too distant future. So in summary, we've got some really encouraging data coming from studies focused on the biology associated with genetic variations of that influence the risk of Parkinson's with alpha-synuclein aggregation inhibitors, LRRK2 inhibitors, GCase activators. And we've also got GLP-1 agonists giving us very encouraging results at the phase 2 stage of clinical testing. So there's quite a lot of activity in terms of disease modification for Parkinson's research. And future bodes well for the field if we can have large multi-arm, multi-stage clinical trials being conducted, accelerating and speeding up the development of all these new treatments. And if you're interested in the general landscape of the field, we put out a paper every year in collaboration with the Michael J. Fox Foundation and Journal of Parkinson's Disease that provides an overview of the pipeline of both symptomatic and disease-modifying therapies. And we're currently in the throes of finalizing this year's version, which will be coming out hopefully in July. And with that, ladies and gentlemen, I'll say thank you very much, and I'll be happy to take any questions.
Simon, can you hear me from here?
I can, sir.
Okay, cool. I'm a bit concerned about the microphones. Simon, I'm just going to step away from the stage and turn on the microphone and see if you can hear me again so we can get questions from the audience. Otherwise, I'll need to think of a different solution. Let's see. I'm gonna, if you have a question, so I'll ask the first question, but so our microphones aren't working, Simon, for some reason. So if people have a question, I'm going to do the unusual thing of invite you up here and stand. Grant has started the line. So please don't be shy and come up here and ask a question. And of course, if you're online, you can either type in a question or we can unmute you and you can speak. So I'll begin. Simon, thank you. Brilliant presentation. Very interesting. I'm just wondering, maybe I'm jumping the gun a bit or jumping ahead a bit with this question, but seeing all these different approaches giving potentially beneficial effects, has there been any consideration at all whether they could be combined for synergy, or is that too soon to go to that stage?
No, that's in-house at Cure Parkinson's. That's a very active area of activity for us. We've got 2 programs, which is number 1, planning for success, which is trying to map out the regulatory process of taking a repurposed molecule through to clinic. If we do have positive results in phase 3, we need to have an active and dynamic conversation with the regulators in terms of what next steps are. You really need an industry partner there, but a lot of these GLP-1 agonists are now off patent, so it's challenging. So that's one area of interest for us. But yeah, obviously, if we have positive results either with GLP-1 agonists or with ambroxol, then we have this wonderful opportunity of exploring combination therapies. And it could be combinations, not just drug plus drug, but drug plus exercise or drug plus diet. So we are currently looking at, we're going to have a workshop later this year, hopefully, or early next year, exploring how others other indications have done this, because oncology's doing combination therapies with everybody. And you've got MS now exploring the space, and yeah, it's the way. It's certainly the way forward, and it's a good problem to have. Hopefully, phase 3 results will allow us to explore it properly.
Yeah, thanks. Grant.
Thank you, Stott.
Thank you, Simon. I'm Grant Jewson from WEI. Fantastic talk. And as a Southampton fan, I hope to be equally as hungover come Saturday, but I'm not entirely positive. So I think the accelerated clinical trials program is really important. And presumably would that come with some standardization in how those trials are set up across different sites? And for example, the lack of biomarkers for Parkinson's is obviously a real challenge, but incorporating that imaging, for example, to complement UPDRS, is that the thought?
Yeah. It's not just it's not just regionally in the UK. There are other MAMS platforms being set up around the world, and what you want is to have universal standards across all of these, so that comparison of data or even cross-platform testing of the same molecule is potentially available, and that would be hugely value added for industry coming in and saying we want to test our drugs on this platform. But yeah, we have a working group in the EJS Act PD program looking at endpoints and standardizing things across all the various centers that are going to be involved. So that's certainly been a major consideration.
And I also want to blame you for the fact that I can't leave Twitter, because you're the only reason I actually stay on Twitter. So thank you, and blame you for that as well. But thank you.
You're welcome. My apologies.
All right, so we'll take a couple of questions from online. Runa's going to help me with this. So, Runa, Anthony Cooper, we're going to unmute you. Anthony, whenever you're ready, can you feel free to...
No, don't do that. Don't do that. Don't do that.
Simon, it's me.
Hi, Anthony.
Go.
Yeah, we can hear you.
Hey, a couple of things. Very quickly, the Australian Parkinson's Commission's second trial is just starting, actually is doing a combination of drugs, just as an aside. But I wanted to ask you in the for ACPD, we'd all love to be able to measure clinical, success or efficacy of a drug. So what is the interim analysis that's going to be used in ACPD at 18 months to make that go, no-go decision?
Yeah, so there's several challenges here. You've got to be recruiting quickly enough that you're going to have enough participants to have a interim period analysis. And the measures that are going to be used are currently being discussed by the working group associated with that, with the end points. So I can't really comment at this point. But okay. Yeah, it represents because previous MAMS platforms that have worked, like oncology,
they've got imaging and biomarkers.
You can take a biopsy, yeah.
Yeah.
It's also a faster turnaround. I mean, there's a terminal endpoint in those cases, whereas with Parkinson's and a lot of these neurodegenerative conditions that are seeking to take this pathway with MAMS, they're very slow, progressive conditions, and the question is, can you actually apply this MAMS template to this nature of disease? So yeah, that's basically the big test.
Yes, exactly.
Of the whole program.
Just very quickly, if I can squeeze in one other comment. Look, GLP-1 agonists, certainly receptor agonists, look pretty encouraging. What's your view or top two thoughts on how it is helping? Which biological aspect do you lean towards as how they may be providing some degree of efficacy?
So Tom Fultonay, who has been the PI on the exenatide work at UCL, has an interesting paper on bioarchive that's hopefully going to be published soon. Cure Parkinson's has been one of the funders on that study, and that's pointing towards potential mechanisms of action. I'll let you have a look at the paper. One of the worries with the data at the moment is that the the data that we have is primarily focused around UPDRS part 3 responding.
Yeah.
And we have post hoc analyses indicating that there is benefits in non-motor scores. But what I am curious about, and I'm off the record here, I hope this isn't being recorded, but I'm curious as to whether this is not just a disease modifying effect, but potentially a bit of symptomatic effect occurring at the same time.
Uh-huh. Okay.
And the GLP-1 agonists are known to elevate levels of tyrosine hydroxylase, for example. So it could potentially be, and they've got a very long half-life. Exenatide's half-life is like 30 hours. So washout is you have to have a long washout period. But I don't think it's ever going to be clean and cut disease modifying versus symptomatic. I think this is going to be the nature of things going forward.
Yeah.
But
Okay.
I'm very curious about that.
All right.
I'm
Well,
not
thank
sure
you.
if I
And back
answered
to
your
you.
question.
Back to you, Wei-Hai. Thank you.
Thanks, Anthony. So we'll take another question from online. So Jeff Stewart, we'll read it out again. So Jeff Stewart asked a question saying, thank you for your presentation. Do you think that genetic profiling of patients will identify personalized treatments for PD patients in the future? Could this also explain why whole PD study groups only produce partial benefit and not whole PD patient groups?
Yeah, that's a really good question. Breaking down the heterogeneity of PD with genetics would be fantastic. Yeah, I think it's a really interesting area to be explored because it is just so heterogeneous at the moment. In each of these studies that we've been conducting, we go back and we look at responders versus non-responders. Who are there any particular details or differences? And I wouldn't be surprised if in the near future we aren't finding some cohorts related to this. So I'd point towards an interesting paper from a group in Bergen just recently looking at mitochondrial analyses, and they found that there is a subset of people with idiopathic Parkinson's that have complex 1 deficiency. That they could be a particular cohort responding to a mitochondrial treatment, but it's lost in a data set full of everybody else. So yeah, it'll be interesting to go back and see if we can't tease out some of this in data from old clinical trials, if that data is actually available in terms of genetics.
Thanks. And we have another question, again from online, from Carolyn Sue. Carolyn says, thanks, Simon. Great to have the overview. Seems that post hoc analyses are finding significant results. Should we be relying on post hoc analyses, or should we be using/designing better primary outcome measures or doing larger studies?
So, hi, Caroline. Hope all is well. The post hoc analyses should always just be considered hypothesis generating. We shouldn't be jumping to conclusions. We have to be careful how we interpret those results. But just as in general, we should be focused on, as a field, we should be focused on better endpoints and primary outcomes in doing larger studies. And hopefully with MAMS platforms, we'll be seeing a lot more of that, or more of a focus on that, excuse me.
Mm-hmm. Thank you. And Ashley Bush online has a question, and actually this was something I wanted to ask as well. So Ashley's question is, what did you make of the deferriprone trial published in NEJM, New England Journal of Medicine? While symptoms were worsened by iron chelation, the volume of the basal ganglia was preserved. Any insights or thoughts?
It was a very early cohort that were tested, and it basically just, well, my personal interpretation is that it just reinforces the necessity of iron in the synthesis of dopamine. These individuals got worse, in the deferriprone treated cohort. But they returned to baseline once treatment was terminated. So I don't think it's necessarily the end of the field exploring iron chelators for Parkinson's. It's just that we have to be careful in terms of who we are treating them with. And deferriprone is an old drug. There are a lot of new iron chelators, metal chelators coming through that are probably better candidates for that testing.
Mm-hmm. Great. Okay, so thanks a lot, Simon, for a very informative seminar. Thanks everyone for great questions and discussion. And we'll wrap it up there for today. Thanks again, Simon. Thanks for getting up early and for joining us for today's seminar.
No problem. Thank you very much.
Bye, Simon.